Okay, so we're online, and hello everyone, and welcome to the spring series of the Electrochemical Colloquium. So in this series, we have an amazing lineup of speakers who will cover a wide range of topics, which are in one way or another connected to electrochemistry. So please check out our website if you want to see the entire schedule for this semester, if you're new to our colloquiums. Now, before we proceed, I want to mention that we often have a rather large audience watching these colloquiums on YouTube, especially today. So they watch it usually either live or in recording. And so for those of you who do so, please make sure that if you like a particular talk, don't forget to click on the like button. That's because by doing so, you will help the YouTube algorithms recognize that this video is valuable and that it must be shown to a broader audience. So today we're going to discuss electron transfer theories that are central to most chemical reactions that we know. And this discussion will be led by Professor Rudolph Marcus, who is one of the founders of theoretical chemistry. Professor Marcus received his PhD from McGill University in 1946. And then he worked at many places, including the National Research Council, which is in Canada, the University of North Carolina, the Brooklyn Polytech, which we today know as New York University, and UIUC. And since 1978, he's been a professor at Caltech. Now, Professor Marcus started his research as an experimentalist, but then during his second postdoc, he decided to explore theory. And sometime later, in 1959, he developed the theory of electron transfer that changed our understanding of chemical reactions and kinetics. So, Professor Marcus, thank you so much for joining us today, and we are really happy to host you at our colloquiums. Now, please, the stage is yours.
Thank you very much, and it's a delight to be here and to discuss with the various participants what's been going on in the electron transfer field, and indeed what's been going on in science. This talk that I'm going to give has a dual personality. It's really a talk in two parts. The first part deals with the many aspects of electron transfer, the many fields that are involved in electron transfer. And the second part deals with a subject which evolved from it, namely the behavior of biological motors. Just how within us and plants, with all throughout nature, does nature succeed after a number of billion years in converting chemical energy to mechanical energy and converting mechanical energy to chemical energy. Several years ago, when the Nobel Prize was awarded for motors, I thought that was great. I will learn a lot. And then I learned that the motors that were developed by man, which were ingenious, were totally different from what nature developed. But of course, nature had several billion years rather than just a decade or two. So let's begin then with the electron transfer, electrochemical aspect of now let me see, how do I get this thing to go forward? Yeah, there we are. The electron transfer field is, when I got into it, began in a very tiny corner of chemistry. And these were reactions that were called isotopic exchange reactions. Here's an example of ferric, and an electron is transferred from the 2+ to the 3+, as you see in that reaction. And the star denotes a radioactive isotope. And the progress of the reaction was followed by looking where the radioactive isotope was as a function of time. And one could do that by precipitating out one of those ionic forms and analyzing the precipitate for the radioactivity. So these reactions were extensively studied immediately after World War II, because there were lots of radioactive isotopes available. And in a way that's interesting because this type of reaction is the simplest kind of reaction in all of chemistry. All that happens is an electron jumps from one species to another, and the products, apart from their labels, are indistinguishable from the reactants. So that means that one of the big factors in affecting reaction rates, namely how downhill the reaction is in terms of free energy, or how uphill it is, how slow it is, that factor was absent. These were essentially thermoneutral kind of reactions. So you could see what's left when that important factor was removed. So this is where it all began, in this tiny corner of chemistry involving isotopic exchange reactions. Here are a couple of more examples. And then later on, there was a study of many reactions that are called cross reactions, where instead of the two species being of the same type, except for their charge, they're quite different species. So the study of cross reactions also became very important. Again, there were examples of electron transfer reactions, but that occurred later, and especially when fast reaction techniques were introduced, because some of these reactions are very fast. In 1952, there was a symposium at the University of Notre Dame in the radiation lab on electron transfer reactions. And there are many, many papers. And in one of those papers, there is one by Bill Libby, who made a very intriguing suggestion. He said that to explain all of these data on isotopic exchange reactions, why some of them are slow and the others are fast, that one should invoke the Franck-Condon principle. Namely, that when an electron jumps from one reactant to the other, it does so quickly when it does happen that the nuclei don't have time to move during that. In other words, he applied what's called the Franck-Condon principle to a chemical reaction. Now, the Franck-Condon principle was originally developed to explain spectroscopic phenomena. And when you shine light on something and the system jumps from one electronic state to the other, to another, an upper state, there's no time for the nuclei to move. And so that helps you in trying to understand the details of that light absorption process. But here, for the first time, Libby applied that famous principle that was developed in, I think, 1927. He applied it in a paper in 1952 to electron transfer reactions to explain why some are slow and others are fast. And the essence of the idea is this. If you have small ions, then there are huge electric fields and huge differences of electric fields acting on the solvent, depending on whether the charge of the particular ion is +2 or +3. And the smaller the ions, the more intense the electric field. And when an electron jumps from its initial environment to the other ion, you now have a new ion present in the wrong environment. In fact, in both ions, they are in the wrong environment for the new charges. And Libby reasoned that the smaller the ion, the larger this discrepancy of of the two types of polarization around the ions, the bigger the barrier. And I happened to be in the library around 1955, 56, and happened to see that symposium volume and happened to see that paper of Libby's. And I was extremely excited about it. I mean, here, this principle, which had been developed for spectroscopy, was applied to chemical reaction rates. And then the more I thought about it, the more I became uneasy. Something didn't seem right. And that often happens, or sometimes happens in science, that you hear about some problem and yes, things look fine. And then you start thinking about it a little bit and you see something not quite right there. And I realized that what was wrong, what was right was applying the Franck-Condon principle. That was brilliant. What was wrong, though, is that when you looked at the details, Libby's suggestion violated the law of conservation of energy. Now, at least in North America, that's a sin to violate the law of conservation of energy, and that's to assume it's a sin everywhere. And how did it violate the law of conservation of energy? Yes, when the electron jumped, the new ions were in the wrong environment, a high energy environment. But where did that energy come from? That wasn't specified. So that's it, the suggestion of Libby, as brilliant as it was, violated the law of conservation of energy. So the question was then, that I asked, was how can one apply the Franck-Condon principle in a way that doesn't violate the law of conservation of energy? And what I realized, that the electron just couldn't just jump from one ion to the other. Rather, you had to have fluctuations such that you reached, let's say a higher energy state. You had to have fluctuations around each of the ions so that the environment around each ions were sort of somewhere in between the initial and eventually the final environment. So if you had those fluctuations to some environment, solvent environment, intermediate between that of the reactants and that of the products, then you could get the transfer of the electron with no violation of the law of conservation of energy. And this is depicted in this slide that I have on there. And you notice this is a slide that Norman Sutton gave me. You notice that in the lower part on the left, there's a +2 charge and a +3 charge. And those little triangles are intended to represent water molecules, and they're more oriented around the higher charge than they are around the +2 charge. And in the products, it's quite the reverse. And so what has to happen for a reaction to occur and not violate the energy conservation is that you have to have fluctuations in all of these water molecules, so it's some sort of roughly intermediate configuration like that. And if you have that fluctuation, then you can have the transfer without changing any energy, and then relax to the products. So that's the essence of the theory. And what I did was actually develop this in a quantitative way and obtain what's called free energy curves of the reactants, free energy curves of the products, and where the intersection occurs is the position along the reaction coordinate, a coordinate I haven't defined here, where the actual electron transfer occurs. So that was the essential idea. And the key part of the theory then was, how do you calculate the free energy, the energetics of that intermediate state, that transient state, which are neither reactants nor products, which have neither the solvation of the reactant nor that of products, but somewhere in between. So that was the key part of the theory, how to do that. And prior to that, I had been involved in a different type of problem, namely one that involves a lot of electrostatics, polyelectrolytes, and their behavior with respect to binding of charges and other phenomena. And these polyelectrolytes are long chain compounds that have charges on them. And these charges interact with the electrolyte around it and interact with each other. And if you have a fairly high salt concentration, the interaction is largely with the neighbors. And a student in a graduate course that I was teaching in statistical mechanics asked me if a topic, if a theory that we were talking about at the time called the Ising model, which treats localized interactions, could be applied to his experimental work on polyelectrolytes. And I thought about it, and well, yeah, yes, it can. And then I got very interested in polyelectrolytes and read extensively on the electrostatics associated with polyelectrolytes, looked at every book on electromagnetic theory that was in the Institute, Brooklyn Poly, where I was at the time, and found one that was the clearest to me. And so I built up a very detailed background in a fundamental way of electrostatics, dielectric polarization, different types of dielectric, their relaxation times, et cetera, et cetera. And so when this problem came up, which is a problem in part of interaction of charges with the surrounding polarized medium, I had, in a sense, the right background to try to tackle this. I had the basics of electrostatics viewed in a rather fundamental way. And the question was how to take that electrostatics, which is normally developed for equilibrated systems, to treat systems which were of this middle type here in this transition state, as it's called, where the polarization around the orientations around the reactants is not in equilibrium with them. So I found a way of calculating the free energy of a non-equilibrium dielectric polarization system, and then applied that theory to this problem, invoked the Franck-Condon principle, and that's how the electron transfer theory came about. So it's interesting, I think, that a key factor in developing the electron transfer theory was this question by a student in a graduate class that I was teaching. And I've found quite frequently, actually, that some of those questions that students ask are really make you think and really do help develop one as a scientist. Well, here are these free energy curves that I came up with for reactants, for products, and the idea is that at their intersection, the reaction occurs. And that delta G0 tells you how downhill the reaction is in free energy terms. The reaction coordinate I haven't specified here, but there's a lot of work done on that. And the expression that emerged then was this simple form, where A takes on certain values depending on whether the reaction has a strong electronic interaction between the reactants or weak electronic interaction. So I haven't written that down there. But this was the key equation that came out of the 1956 theory. In the lambda there, which is called the reorganization energy, it's broken down into what's happening into the hydration shell around each of the reactants and any other vibrations that are around, and a contribution that's related to the radii, the size of the reactants, those are the A's, the distance apart, and the difference of the optical and static dielectric constants. It's this thing here where that non-equilibrium polarization comes in. And one can understand all of this in physical terms, which I won't have time to go into here, but be glad to answer any questions. Now, in the theory, I had a choice to make between presenting very complicated equations or making some additional approximations that really simplified them. And in particular, these F's here are force constants, and you notice they're symmetric in the force constant when it's a reactant, and when it's a product, it's a symmetric expression. But that meant that I neglected, and this is in one of my articles, and I specified what they were, the asymmetric terms. And the question was, yes, this formula is less accurate than the other one would be, but the other one, well, I'll come to it in a moment, is such a mess that I don't think experimentalists would have used it. Well, from this equation, many things came out, and one of them is called the cross relation, whereby you take two isotopic exchange reactions, measure the rate constants, and if you know the chemical equilibrium between them, which you often do, that's the equilibrium constant K, you could make a prediction about what the rate constant would be for the cross reaction. And this was the expression that came out. And so, and this F here is in many cases close to unity. And so the question was, as one of the applications of the theory, how well did that work? I mean, you measure the two isotopic exchange reactions, you measure the equilibrium constant of the two, between the two types of systems, and you predict the rate constant of the cross reaction. How well does it agree? So that was one of the early applications of the theory. And here's a slide taken from a review article by Norman Sutton and me in 1985. And the details don't matter, but this gives you an idea of the variety of self-exchange reactions and the cross reactions. If you look at the article, don't look at the slide, you'll see that the rate constants vary by about 7 orders of magnitude. And, you know, the agreement isn't bad. The agreement's pretty good, considering that there are a number of effects that aren't taken into account related to the charge effect. So that was an early example of an application. Then there were all sorts of consequences of the theory. Supposing that one considered a system which was very, very downhill in free energy. In other words, where the products curve of the cross reaction was very low compared with the other. Well, you see what's happened. If the products had about the same free energy as the reactants, you'd have that sort of a curve, and you'd have an intersection there. If you consider a series of reactions where you systematically, by chemical choice, make the reaction more and more downhill, eventually the products curve intersects near the bottom, and you have what's called an activationless electron transfer, which has been observed. And then if you go further, the barrier starts increasing again. And so I realized this in a paper in 1960 and called it the inverted effect. And this was one of the predictions. And this was actually the prediction that took 25 years before it was confirmed. There are many, many attempts to, but nature always found a way, if the reaction became slow due to this, to find another chemical route for making some reaction occur. And then finally, in a classic paper that's been well recognized by John Miller, and a combination of a study of by a physical chemist and an organic chemist and the student who did all the work. And this was the system that was constructed by the organic chemist. Here's a reactant, and here's the other reactant. And there's no chance that they will come close to each other and so have one of those other reaction mechanisms. And this is a rigid bridge that separates them. And sure enough, they got the inverted effect. Oops, that went. So this was the first example, and it was a life-changing. Well, it was a field-changing and life-changing experience. And since then, there have been many examples of the inverted effect. But it took, this is one that took 25 years before there was experimental confirmation, even though many people tried. And the secret was keeping the two reactants far apart so they didn't undergo other types of reactants that hadn't been anticipated. There weren't electron transfer, they were atom transfer. Here's an example. A strange thing happened at this point. The theory had been developed for electron transfers, and the cross relation was developed for electron transfers. And then Norman Sutin applied it to a reaction that he shouldn't have applied it to, an atom transfer reaction, and it worked. And so that sort of set the stage, I guess, for other people to apply that cross relation to reactions that have nothing to do with electron transfer. And here's an example by Lewis and Hugh from 1984. And here's an example. It's an aryl sulfonate, substituted aryl sulfonate, reacting with a methylated form. And here you have a transfer of a methyl group from one reactant to the other, not a transfer of methyl group. And here's the cross relation. The theoretical curve is the straight line, and the experimental points are the circles. And there's no adjustable parameters in there. So this was an example following Norman Sutin's application to a different kind of reaction of the cross relation to a different reaction. And here's another one that comes from a review article by Jim Mayer and a student. It's published in, and this was transfer. This was a hydrogen atom transfer, and again, the points are the experimental points, and the line is the theoretical line for the cross relation. And here's, so not only was the cross relation seen experimentally in reactions that had nothing to do with electron transfers, but they were also seen in computations. And here's an example of a computation. Again, the points are the computed points for the cross reaction and its behavior, and the line is the theoretical line, that simple formula. So it's an example where theories have been developed for one type of phenomena, but there's something in it, inside it, which it might be common to other kinds of processes. And if you notice the cross relation, there is nothing in that cross relation which had the specific properties like the dielectric constant, or it just had rate constants and equilibrium constants. And when you obtain a formula like that, then that means it may be more general than what it was derived for, because it doesn't depend explicitly, or need not depend explicitly in the equation form it's given, on the parameters that are specific to the problem for which it was designed. So it can be applied to other problems, hopefully, and you see it works, or it worked. And there are other results of the theory. One is the, how are electronically excited molecules produced in a chemical reaction? In the theory, if the system is in a very downhill region, there might be an excited state which will intersect the reactance curve more readily. And if you form that electronic state, then in various ways you can get light emitted. This then is an explanation of electrochemical luminescence. And the electrochemist Alvard was the one who discovered it. This electroluminescence is a result of electron transfer in the 1960s. Let's see. Why don't we go for it? Okay. Now, there have also been simulations, quite a number of computer simulations to test the theory. And let me see, this is, I think I have an earlier one than this. Let me just see if I do. No. Oh, yes, someone. Yeah, there were, I skipped a few slides. Let me come to that. One of the problems in electrochemistry that was pursued a lot in the 50s, 60s, 70s, was to try to look at how the log of the current depended on potential, the so-called Tafel plot. And in the theory, a curvature was predicted instead of the usual straight line that's involved, for example, in Butler-Polanyi theory. So could one test that curvature? Could one test that that result that I showed for a particular reaction, where there was a real pronounced curvature of a plot of log K versus the standard for energy. In fact, it turned over. Could one see something like that, although no turnover because the special properties of the electrode? Could one see that in electrochemistry? And many people tried to look for curved Tafel plots, the log of current versus overpotential, and completely unsuccessful. And then in 1991, Tsitsi tried a different tack. He built a monolayer on top of the electrode and attached a redox agent, a ferrocene, to one end of it. And now one of the factors that plagued the electrochemists at the time, namely that if you try to investigate fast reactions, and some of these became fast, that the slow step would be diffusion, and they just couldn't observe that curvature. Well, Tsitsi arranged things so there didn't have to be any diffusion. And then he went ahead and observed the curvature and applied the theory and what have you. So some of these things take a long time to appear after the development of a theory, including some later than this, but they're welcome nevertheless. Then there are the computations. There have been a fair number of computations of these free energy plots of the reactants and the products. And one of the, perhaps the earliest one, one of the earliest, was by David Chandler and co-workers in 1988. And here, taken from his paper, is this plot. And there have been others, Tom Miller and co-workers, and Manilopoulos and co-workers, and other. These have been some of the leaders here, and others. And here's an example from a paper of Tom Miller, where the dots are the computer results, and the others are the theory, and you see how closely it managed. Now, this is when the results are largely classical, when you bring in quantum mechanics, as some of the, as Tom and as Manolopoulos did later on, then you will get quantum effects which are not described by the simple theory. All right, this is the short break time. This is sort of the, as much as I thought I had time to say, and on the electron transfer part of the talk. And the next part of the talk will be on these biological motors and how the work on them was stimulated actually by the success of the cross relation in those reactions that had nothing to do with electron transfer. In other words, what do these motors have in common with these other simpler chemical systems, and also how is chemical energy converted to mechanical energy and vice versa. So maybe we can take this time to, if there are any questions, I'd be glad to try to answer them.
Thank you, Professor Marcus. So we are open to questions. Is anybody have, you can raise your hand and I will unmute you so you can ask directly, or you can ask in the chat. Maybe I can start in the meantime. So one question was about the quadratic dependence of free energy. And I think in general, one can probably rephrase it and put it in a different way. So when do you think that this quadratic equation dependence really breaks down? For what cases? So when the nonlinearity really comes into play in the theory, or should be considered?
Well, there are several answers to that. One is when you include nuclear tunneling. And if you look at the inverted region, the barrier to tunneling is like almost like a moon. The barrier to tunneling is much narrower than when you look in the normal region. In other words, where they intersect with slopes of opposite sign. So the nuclear tunneling will produce an asymmetry. And the effect isn't large. Well, it depends what you're looking at. But if you recall that slide that I showed of that long-range electron transfer and the reactants being kept separated by a bridge, there's an asymmetry there. And sometimes that asymmetry has a big effect. So essentially that you might call a breakdown from the quadratic behavior. There are more complicated expressions. There's one that I derived and is in Appendix 4 of the 1965 J-Chem Phys paper, which doesn't just use the symmetric form of the combination of the force constants, but brings into account the correction due to asymmetry. And if you look at the formula there, that's in Appendix 4, it's a long formula. And I think practically no experimentalist would be interested in using it. I mean, it's obscure, it's, but nevertheless, it's more correct. But I made the choice, for better or worse, to put it in an appendix because it looked so complicated, even though it was more correct. And in a sense, and I say this to the young people, there are some things maybe one shouldn't bury in an appendix, because a number of times there's been some criticism, occasionally some criticism of the literature, for taking the force constants, corresponding force constants in the isotopic exchange to be the same, whereas they're actually different. Well, in that 1965 paper, in that appendix, I have the correction for the asymmetry, but only a few people have noticed it. Recently, Richard Compton actually observed that asymmetry in some cyclic voltammetry. The Tafel slope is not quite 0.5, and indeed, that deviation from 0.5 at zero over potential is present in the Appendix 4 of the 1965 paper. So there is an example where it took many years before there was finally an observation which brought in specifically the role of the asymmetric force terms.
Thank you. Yeah, I guess the theory took a long time to confirm. Another question.
Certain things were confirmed right away. And
Right.
So there is immediately recognition. The number of invitations at talks grew tremendously. But there are certain aspects of the theory that took decades. In fact, in one case, I think 4 decades, for the
inverted region.
For the experiments to realize there was a theory there.
Okay.
To explain them.
We have a question from Lior.
All right.
Hello, a quick one. Can you hear me first of all?
Yes, perfectly.
You showed an example where the methyl transfer also obeys the Marcus theory. And it's a bit surprising if we think about the rate of electron transfer events versus methyl transfer events. Can you maybe explain how come this still holds where we think about the picture of Franck-Condon as well?
Yeah. I too was surprised at how well the electron transfer theory was working for reactions that had nothing to do with electron transfer. And so in 1968, I wrote a paper where I used some simplified expressions that were available due to Harold Johnson and Linus Pauling on the energy of a bond and the energy of a forming bond, the energy of a breaking bond. And the essence of that theory, which wasn't mine, the essence of that theory is that the bond order, whether it's a single bond or double bond or something, or no bond or something in between, that during the course of the reaction, as one atom is approaching, or as one group is approaching another and another group is leaving, the assumption was that the total bond order was constant. You break one, you form another, and you have that kind of compensation is constant. And so taking formulas that have been developed by others for the energy of a system with bonds broken and formed, I then made some approximations, the mathematical approximations that were fine, they were okay numerically, and derived a formula for the free energy barrier and derived a formula then for the cross relation. And the formula for the cross relation was almost the same as the one for electron transfer, except in the factor that is close to unity, often, namely what was F12 in an earlier slide that I showed. So that part was different. And also for these other kinds of reactions, there is no inverted effect. The physical reason for the inverted effect was missing. And so the cross relation then can be understood in this very approximate way with that kind of model. And that model is introducing nothing about electron transfer. So that's one answer. Of course, it would be good, and nowadays with computers, one might be able to do that, and plus formal theory, show by looking at groups of things, partitioning things, and so on, that one can derive the cross relation, at least for standard free energies, which are not too large compared with the reorganization energy. One can derive that cross relation approximately for all sorts of reactions. And certainly experimentally, it's been observed. You see. So that's always a encouraging thing for the young people in the audience, I always like to suggest to the students, please look at the experiments. They're the ones that are a result of nature being around for a long time, and they're a rich source of unexpected phenomena to try to explain.
Thank you.
So then, where would you say that the theory breaks down in a briefly? Is there any?
Yeah, that's a good question.
Or maybe it doesn't.
No.
So one of the reasons why I'm asking is because we also had a discussion previously. You probably attended the talk.
No, I'll give you an answer. I'll give you an answer. Supposing you go to ultra-fast reactions that take place almost within a vibration. And you know, there are reactions that are being studied now, we're at 100 femtoseconds, so on. And they're reactions at surfaces, electron transfers at surfaces, which seem to maybe take 20 femtoseconds. Now, how much of that is a charge transfer spectrum rather than actual excitation followed by electron transfer is another question. But certainly in the area of fast reactions, eventually, there isn't time for certain things to happen. So one would expect that there, one would want a modified theory, a more quantum mechanical theory. I mean, this theory has lots of quantum mechanics in it, especially contributed by others. But certainly in the field of the ultra fast, where it may be that they don't follow a kind of a simple exponential behavior, a decay or something. In other words, the time evolution may be more complicated. So I think that area and ultra-fast reactions are increasingly being studied. At one time, ultra-fast was nanoseconds, then ultra-fast became picoseconds, and now ultra-fast is femtoseconds. So the definition of ultra-fast changes too. But I think that's one place. Of course, the theory has many aspects to it. So some parts, for example, the part, it's not necessary to use dielectric continuum theory, and indeed in the 1965 paper I didn't. But if you look at various versions of the theory that put in specific models, then of course, when there are specific solvent effects, that part will break down.
Would you say that if we treat heterogeneous electrocatalysis that we discuss often with this theory, we may run into also some inaccuracies?
Well, yes, because first of all, the whole situation may be far more complicated. If the reaction is occurring at some edge, say, in some on a crystal surface at some edge boundary or something, well, those are details that aren't part of the theory itself. One makes a theory for that. So there are all sorts of specific effects that one can imagine where one wouldn't apply a simple theory. One would actually look at it and see, well, what is the evidence for how the reaction is occurring, and how can I imitate that by theory? And certainly items will come in there that aren't involved in the simple description that I gave earlier.
Thank you. This is a very good point. I think that, Professor Markham, we can continue.
Okay.
At this point. Yeah.
All right. All right. Here's where it all began. This is a bilayer membrane, and this describes the photosynthetic system where you have light harvesting compounds that pick up light and that eventually transfer their energy to this localized reaction center, and a series of electron transfers occurs, and where proton transfers occurs, and all sorts of things happen. In here is the part that I'm going to be talking about, the ATPase, which is responsible for formation of adenosine triphosphate, which is used for many processes in humans and plants, in life in general. And in here are, this is the part I'm going to be talking about here, which is involved in transferring protons across the membrane and in using adenosine diphosphate to form adenosine triphosphate, which is used to do various things, or vice versa in other systems. By the way, in this diagram, there are at least 4 Nobel Prizes. So if you want to see where some of the prizes occurred, they're in this diagram here. One of them is for the structure of this photosynthetic reaction center. Another is for the structure of part of this ATPase. Another is for the mechanism for transport of protons across the membrane. Another is for some electron transfer, and there may be others hidden in here too. Melvin Calvin may have some place in here, I'm not sure. So anyways, this I'm going to be talking about part of this ATPase, and the part of the question there was over the years, there have been many chemical studies of enzymes and reactions involving ATP formation or what have you. And in more recently, past 20 years or so, there have been studies at the single molecule level. And then they have revealed all sorts of things. And I was intrigued by some of the things they revealed. And then with a wonderful collaborator, Sander Volkan-Katsko, who's very brave in tackling all sorts of things in biological system, although he's a physicist who was brought up in single molecule studies of quantum dots, totally different field, has done. So that's the second part of my talk then, is about single molecule studies related to that ATPase. Here's, let me see if it works. Yes, it does. Here's an animation from John Walker's group. He did the structure of part of that ATPase. And what it's intended to show is ADP going in to various clefts, and ATP coming out from other clefts, and the various motions, large-scale motions of these protein subunits that actually permit openings of channels and closing of the channels, and permitting reactions to occur when they're closed and so on. So nature has evolved this process, and this is a reconstruction from structures that have been found with various substituents of the model, and from that, Walker created this model. So that's, Now, always, if you enter a new field, you should have something to present that's different from what had been done make, in order to have some interesting results. And our entry into it was in adapting that cross relation to this problem here. Now, we saw in electron transfer, there is sort of fluctuations around each of the reactants so as to get compromise configurations that permitted the electron transfer to occur, satisfy the Franck-Condon principle, and so on. And in the case of the transfer of methyl groups or methyl plus groups or other groups from one reactant to the other, is a somewhat different story. There, at least in one model, it was that sort of approximate conservation of bond order. You form bonds and you break bonds, and the forming bonds helps the breaking bonds to occur. And in fact, it reduces the barrier by about to about 10% of what it would be if you didn't have that kind of cooperation. And so in this case, it occurred to us that when an ATP enters one of those clefts, you remember it goes into a sort of a cavity, and when it enters one of those clefts, of course, it's interacting with the clefts a lot, and as it moves in, you're breaking hydrogen bonds, you're forming hydrogen bonds. And so instead of breaking and forming ordinary bonds, you're breaking and forming hydrogen bonds. And so we assumed that maybe it obeys that same sort of quadratic free energy expression, approximately. And so that was our entry into the field. Let's see how far we can get with using that expression. And can we use it in a way that brings into the question, how does chemical energy convert to mechanical energy and vice versa? How does it do it in nature? So that's how we got into it. There was different, as I understand it, from what had been done before, although there had been a lot of pioneering work done before, particularly by Oster. In these single molecule experiments, what they did was they took the ATPase and they cut it. They separated the part that was outside the membrane from what was inside the membrane. And then they took that part that was outside the membrane and stood it on its head on a slide and glued on a slide, and then attached a monitor, maybe it was a rod, a nanorod or what have you, a monitor to that central, there's a central shaft, and they attached to the central shaft, and they followed the progress of the reaction in part by looking at how the central shaft rotates when you do various things. They also followed it by taking the ATP and adding to it, attaching it to something that is luminescent, the fluorescent, when it's bound to the ATPase and doesn't fluoresce when it's in the liquid. So there are several ways then of following the reaction, both of the single molecule, both optically and by fluorescence, and optically actually by looking at the scattering as it's rotating. That has nothing to do with the fluorescence. So there are different methods of studying it. And the key question was to take this rather artificial system where you cut it apart and see, does it obey the same rules as the system which isn't cut apart, the actual system in solution, if you run it that way. And these early experiments are designed to show that, yes, they do. The experiments done in 2001 and by and large, the curves overlap each other. And let's see, here, one of them involves this nanometer bead, what have you. And so there's that part which is satisfied. So the work that was done, it was argued that the work was done with single molecules, the truncated system was relevant to the actual problem, even though it couldn't perform certain functions. Among the types of experiments, there are two types. There are any number of types, but one is here's this the part of that ATPase, the part that's outside the membrane, stuck on a slide, and it has this central shaft that's made to rotate. It can rotate. You can control the rotation by magnetic methods. You can look at in other experiments, you can look at the fluorescence. And the idea is that at different rotation angles, the free energy curves would look different. And you would take that into account in the theory. And so the one question was, can we take some of these experiments obtained by one method and use the theory, that quadratic expression, to make predictions? Here's an example of these are controlled rotation experiments, and using data from other independent experiments, we obtained the red line and these various other lines. Some of them are for a rate constant, one is for an equilibrium constant. And the points are the experimental points, and the lines are the theoretical lines without any adjustable parameters. So that's an example of what one can do with that cross relation. Now, I wonder if, I'm looking at this, whether I've skipped over some slides. Yes, I think I have. Yeah. All right. Here is a slide that I skipped over. How does the connection between chemical energy and mechanical energy come in the system and in the model that we're using? In other words, what is the connection? And the idea is this: when that central shaft rotates, there are certain local equilibrium positions that are found where it would tend to be stable for the moment if it didn't have too much momentum. So you get fluctuations around those local stable positions. So the mechanical part then in the theory comes in writing down expressions for these fluctuations when it's in the reactant's local stable point compared with when it's in the product's local stable point. And these are angles associated with those points, and theta is the existing angle. And actually, this kind of behavior is observed from optical experiments by looking at fluctuations in angle and seeing that they obey that kind of behavior. So the mechanical energy comes in fluctuating from stable positions that are mechanically different. And then the free energy of activation involves this formula that we've seen before. And now these work terms are these mechanical terms here. And then one can get the standard free energy reaction expressed in terms of them. One could get the rate constant expressed in terms of it. So that's the central idea, that we connected the mechanical and the chemical by looking at these stable points, looking at fluctuations from each, and the idea that you get reactions from going from one of those parabolas to where an intersection is, and then going over to the other. And you do that at each angle. So for different angles, you have different parabolas. The relative stability of products compared with the reactants depends upon the angle of rotation, the degree of openness of the cleft that the ATP or ADP went into, what have you. We speak then of a standard free energy reaction at any particular angle, and that varies with angle, and so on. So this is the way we sort of combine the energy, and then this plot you've seen. Interesting enough, there's something, as you know, in electrochemistry, a Tafel slope that describes the current as a function of the overpotential. Well, in chemical reactions, there's what's called a Bronsted slope, which again is a kind of a logarithm of a rate constant versus how the driving force of the reaction, the standard free energy reaction, how downhill or uphill the reaction is. Well, in these systems here, in one slide that I showed you, it showed a good agreement, and there the Tafel coefficient or the Bronsted coefficient was about 0.5. But they also have reactions related to the hydrolysis different step, where the Tafel slope or the Bronsted slope is 0, and the opposite, one other type of system, where the slope, the reverse reaction, where the slope is unity. So this motor is rich in terms of Tafel slopes. There are reactions for which is 0, 1, and about 1/2. And that's interesting, because when you look at detailed structures, it becomes an interesting question. Just how do you account for these Tafel slopes of 0 and 1? And it's still an open question, although there are suggestions that have been made, for example, by area Warshel, so on. But it certainly is something that we're going to work on in the future and try to understand that better. There are things one has to be careful about, though, in working with these proteins. There are some that are designed to work at high temperature, and when you study them at room temperature or lower, maybe substantially lower than room temperature, they sort of freeze up. They give poor behavior. And then if you try to interpret the effective temperature, you get abnormal results, normal frequency factors, and so on. And here is some study where they plotted the ATP rate versus 1 over temperature. And here there's a break in the slope. And so we think that for simple interpretation, only those, at the high temperature, and that below, it's a different system. And in fact, when you calculate Arrhenius factors, you get some ridiculous result for the Arrhenius factor in these regions here. And that's been noticed before in systems that have nothing to do with ATP, but a different kind of enzyme. Judith Klindman has studied it, and where you have really abnormal behavior at low temperature. So one has to, in analyzing some of the data on these proteins, if there's any that's at lower temperature, you may get results that maybe reflect more of the glassy nature of the protein than they reflect anything about the chemistry of the phenomenon. Here's an example of some data in a controlled rotation experiment. And these are looking at fluorescence when it goes in, when in this case, an ATP molecule goes into a cleft, then it starts fluorescing, compared with when it goes out, then it stops fluorescing. And these dots are related to observations on that. Now, you'd think from that, it'd be impossible to come out with some sort of simple curves or what have you. But Shandor, collaborator, is mastered handling these situations, and with the aid of a student who worked on it, this came out of it. This shows, this isn't the final slide, the work is still to be done, but it shows a correlation between the ATP binding and 240 degrees later, ADP coming out, and they're closely correlated. And in fact, in nature, when one studies these things, these systems, it's been found that if you have a low ATP concentration, then it's hard for an ADP molecule that's inside to come out. And that you can accelerate the rate by at least 1 million times by having a high concentration of ATP. And so that there's always an ATP going in when the ADP comes out. And there's an example of a concerted motion. And that's one of the things that we want to try to understand. And maybe sort of qualitatively, you can understand it in the following way. Imagine that you have a ball and that you have clefts in that ball. And that if you close one cleft, you're going to open another. So if the one that you're closing on is the ATP going in and reacting, and the one that you're opening up is the ADP coming out, then you can see there's going to be a correlation between the two if those events can happen almost simultaneously. And this slide, or whatever replaces it when we finally do the last part of the calculations, shows that strong correlation. Now, it's been known for quite a while now from work in Japan that there is a correlation, and that it can have a dramatic effect. You have this huge catalytic effect. Oh no, I went too far. Yeah. Nevertheless, in textbooks, here's one. You have mechanisms which was okay before the single molecule data, but it has the ADP PI coming in as a separate step from the ATP going out. So at least in this book, one would actually combine these things, one would make them more simultaneous. But that's a little example of how some of the single molecule studies can affect what's known about mechanism. Well, it means to, to acknowledge my collaborators. One is Sandra Vogan-Katsko, who's been wonderful, and we've written quite a few papers together, and he's brought in a number of his students from Azusa Pacific who've done some of the work we described. And then there's Ricardo Matute, who is a professor in Chile, and he is heavily involved in these studies also, and contributing a lot. So this is the group working on the ATPase. It also, it's fun to interact with people, and I've enjoyed very much my interactions on other problems that are totally different from this. Kai Yu, Wei Chen, and Jacob Soa. of course, there's some financial support, the Office of Naval Research, the Glanville Fund, and the Army Research Office are the most recent ones. Well, thank you very much, and be glad to answer or try to answer any questions that might arise.
Thank you so much, Professor Marcus. I think we're open to questions. So again, you can write them in the chat or you can ask directly. We have a question from Raphael.
Hi, thank you for your talk. I wanted to ask if there is any advice you would give for someone who is starting a PhD or just a young researcher in general.
That's a good question, a very good question. And of course, it's hard to give an answer, in part because there's so much pressure to get results done nowadays. The support, there's a lot more pressure than there was when I was a young student or postdoc and so on. But I can only say what worked for me. But it's a different age. What worked for me when I look at the various types of research that I've done, of which electron transfer is one of them, but just one, that it's often being guided by what the experiments are available and what they're telling us. Nature has had billions of years to develop phenomena, and there's all sorts of surprises that we don't think of. So keeping an eye out in looking at the literature for, what seems to be interesting, what seems to be new. But then also, and this I wasn't conscious of at the time, but as I look back, I've always tried to link the dots. In other words, for example, in the problem, a non-water catalysis that came to my attention, whereby you can get maybe a factor of million increase in rates of organic reactions, certain ones in an emulsion. And there didn't seem to be an explanation. And then I remembered, well, in some frequency generation experiments, at the surface of a water, you have OHs sticking out rather than sticking into solution. And if they were more attached to the transition state than the reactants, you could get huge catalysis and so on. So we were involved in that with Yusun Zheng, who made some calculations and so on. But that was sort of a matter of connecting the dots, or various things that happen in the stratosphere of the ozone composition up there, where there's some anomalies in the isotope effects, and linking that dot with things like degeneracy of rotational states and getting chaos and unimolecular reactions and so on. So in my own case, as I think back, without being conscious of it, a lot of it has been the matter of connecting the dots, but certainly a lot of it has been listening to things or reading about things, and being on the lookout for something that seems new. Now, there are a lot more people in the field now. It's probably a much more difficult task now. The competition is probably more now. But nevertheless, the number of results are available is much more now. So there's a better chance that way. But I don't know that this is advice that I can give, because at the same time, you have to bear in mind the pressures of having to publish in order to get or renew grants. But certainly one thing I can certainly say, don't think about prizes. I mean, it never occurred to me to think about a Nobel Prize, anything like that. I think it might have interfered if I had with the research I was doing. might have taken some of the fun out of it because the chances statistically are so small. But I think just curiosity about understanding things, if one has the luxury of being able to do that.
Thank you.
Thank you. Ben, you can go ahead.
Hi, great talk, Professor Marcus. I really enjoyed it. I guess switching back to a scientific question, I was hoping you could maybe talk a little bit more about the symmetry of the barrier. You spoke of the
Oh yes, sure.
the different Bronsted slopes,
Right.
And how they take different values from 1/2 and all the way up to 1.
Oh, that, yes, sure.
Yeah.
No, that's a very interesting question. And for this particular problem, there's research still to be done to try to understand it fully. We have various ideas, we're working on them, but we don't know if they're going to pan out. But the essential idea is this, the broad theme is this, that if you think of the reactants, and if you think of the transition state in between, in the case of electron transfers, where those parabolas intersect, and if you think of the products, then if the intersection is close to where the reactants mainly are, namely the bottom of its well, then essentially changing the free energy is going to have very little effect. You move the one parabola horizontally to the left or right or down, it's not going to change things. And that'll correspond actually to a transfer coefficient of 0. If, on the other hand, you move the parabola so that the reactance parabola intersects near the top of the, intersects the products one at the bottom of the products react, then the transition state, where the point where the two things in, is very much like the products. And then one expects a Bronsted slope or a Tafel slope of unity. And if, as is the usual case, it's sort of somewhere in between, and sort of halfway in between, then you would get a Tafel slope of 0.5. So a key thing then in understanding those Tafel slopes is understanding the relative positions of the reactants and the products free energy curves, as well as certain other factors that come in that I haven't mentioned. So it boils down to the mechanism, and the mechanism is not simple. I mean, in the case of ATP, where you break a bond to form ADP, an inorganic phosphate, you have attached to the AT a magnesium ion, which is attached to 2 phosphates, and then after the breakage, it's attached to 2 other phosphates, one of which is in common. So it's moved quite a bit. And I've looked a little bit at computer simulations, and by and large, as far as I can tell, they don't really go into those kinds of details. So I think there's much to be understood yet in for example, in computer simulations. One of which. Pardon?
Oh, no, sorry. That's great. Yeah, thank you. Maybe just a quick follow-up question to that. So in electrochemical reactions, this transfer coefficient is often assumed to be about 1/2. So is that a safe assumption to make? Is it based on
well, if the overpotential is close to 0, in other words, if the 2 wells have an energy about the same amount, a free energy about the same amount, so you have 2 parabolas and their bottoms are the same energy, then that's probably a reasonably safe assumption. There's a correction for an asymmetry force effect that I mentioned that's buried in Appendix 4, and that Richard Compton more recently has found experimental evidence for, but it's a small term, but nevertheless it's there, and it should be there, but that would be a deviation, but that wouldn't be a large deviation. If there's different mechanisms coming in, more complicated mechanisms, then of course that's going to change things too. But for simple electron transfers, I think that's a fair thing. I mean, there is, of course, the nuclear tunneling contribution that is not taken into account in the simple theory. On the other hand, in computer simulations by Manonopoulos, where he had quantum effects, he found that you can really imitate things if you use a somewhat different lambda. But, that's sort of a bit ad hoc. Yeah, anyways, that's
great. Thank you very much. Yeah. Thanks.
Thanks, Ben, for the question. I can also refer to our previous talk by Professor Elizabeth Santos in December, so where we discussed these things in more details as concerned the heterogeneous catalysis, electrocatalysis.
Yeah, by the way, I noticed that one of the future talks is by Proton, is by Sharon Hammes Schiffer, and there too, Jim Mayer observed the inverted effect for these electron proton coupled transfers. So some of this may arise there too. Yeah.
Yep. So I think that maybe we'll have another 10 minutes at most for the questions, if it's okay for you.
Oh yeah.
So one question that I have, still staying on science. So you said that you used the theory with the quadratic expression for the energy curve in the calculations of the ATPase, right? So one question that I have is, if you use it, does it mean that you can you feel that it's safe to ignore any nuclear effects, any proton tunneling?
Yeah, no, you have to superimpose proton tunneling. But let me make a comment on that quadratic business. What I did really was wrote the free energy expression and the free energy of the fluctuations in terms of the standard dielectrically unsaturated approximation. And that gives rise to a free energy thing. What I didn't realize I was doing, just simply didn't, is that I was using a statistical mechanical expression, and I was using what's called the cumulative approximation in that, whereby one takes the average of an exponential, a Boltzmann factor, and relates it to the exponential of a sum of averages. I was using a cumulant expansion and stopping at the second order term in the cumulant expansion. So it's interesting that it's like, I think it wasn't it some chap in one of Moliere's stories where he didn't realize he was speaking prose all the time. So actually, without realizing it, by using the free energy expression and then using that, it was equivalent to in statistical mechanics of using a cumulative expression expansion and stopping at the second term in it. It's interesting.
Okay. Yeah. So maybe I can ask another question, and there will be the final one from the chat. So we see in theory, if we look for the last, like 20 years, there's a considerable rise of the use of DFT and molecular dynamics simulations for all sorts of problems, just like a brute force solving, calculating, getting the quantities out, and that's it. So how do you see that sort of progression in terms of the theoretical chemistry and in terms of the progression of the field, of the community? Is it something we should be careful about, or we should not be too much afraid of going into pure simulations without really developing the formalism, the theory.
Yeah, that's a very interesting and far-reaching question. And by the way, it occurs not only in chemistry, but it occurs in all fields. Aeronautic. I remember I often had lunch at a round table where there are a number of engineers, and that same trend has occurred in aeronautical engineering and other fields too. That computers have increasingly taken the whole. Now, of course, that has several consequences. First of all, with the computer calculations, you can address certain questions that we couldn't possibly address years ago. I mean, not just the electron transfer field, any field. Unimolecular reactions, for example, that I've been heavily involved in. With the computers, you can remove certain assumptions and see what effect they have. You can evaluate the assumptions. Certain things like electronic structure and potential energy surfaces, of course, there's no other way, really. I mean, there all sorts of approximate ways were tried in the pre-computer days and were very helpful, but there's no competition with modern computer ways. Of course, there's a lot of room for ingenuity within that computer framework. But so for addressing certain questions, computers really have added a lot. On the other hand, in some cases, it's changed the nature of some people's research. Their research then, in some cases, is not related to the real world, but how can I get this result faster and more accurately? Though that's an important question, but it diverts attention from the real world. So problems that one might be looking at may not have been stimulated by experiment, but just simply because the person involved thought, well, what if I try this? What's going to happen? Or something like that. And there's certainly been occasions where computer results have stimulated development of analytic theory. I remember many years ago seeing some computer results by Wall, Porter, and Karplus on a chemical reaction, H plus H2, and how vibrational energy could help that chemical reaction go. And when I looked at their data, I saw that their computer data, I saw that I could explain the results by assuming that even though during the course of the reaction, the HH vibration changed considerably from one involving just H2 to one involving H3. This is an H plus H2 reaction. That if you assumed it was in the same quantum state during that whole thing, you could explain the results quantitatively. And then I saw some results by Morton Pitzer that did the H plus H2 collinear reaction on a computer in quantum mechanics. And I saw if I made the same assumption, I could explain their results. And so then I developed what later be called vibrational adiabatic theory as one type of chemical reaction. But in that case, it was seeing computer results that simulated the theory. Of course, much later, I found that in the early 1930s, other people had thought of it before computer age. But, it's an example of where computer results can, well, another example is in Bernie Alder and Wainwright's study of hard sphere fluids on a computer, where they found these long time tails in the computer, and then people were able to explain it in terms of hydrodynamics and what have you. So, sometimes things are discovered by computers, and I imagine there are many other examples. I'm just familiar with those two. But the question is, it's important to have the computations. It's important to have computations which provide insight. There is the danger, there always is a danger in any kind of research, that one may become involved in things which have nothing to do with the real world. And maybe that's not so bad, considering the real world, but it's, I'm not sure how much that helps science.
Okay, thank you. Thank you so much, Professor Marcus, for this excellent meeting and excellent talk. So I think that probably at this point we will we will end. And I see more questions, but as usual, we cannot just keep asking questions forever. So with this, thank you so much.
I will thank you, and I've enjoyed interacting.
Thank you.